You're listening to Conferences Online Allergy from Children's Mercy Hospital in Kansas City, Missouri. Today is October 21st, 2024. Our topic is Abbas Chapter 3, Leukocyte Circulation and Migration into Tissues. This is presented by Dr. Nikita Raje. She is an Associate Professor of Internal Medicine Pediatrics and is the Chief in the section of Allergy Immunology.
Few disclosures here, nothing relevant to the talk. And today's learning objectives are to recognize the types of adhesion molecules and to learn the pattern of migration of various types of leukocytes. Couple of questions here, we'll come back to these at the end. Migration of leukocytes out of the blood into the tissues mainly occurs in which type of vessel? And which of the following statements correctly describes the migratory behavior of naive T lymphocytes? Right. That's the outline for today's talk. Let's start with overview of the leukocyte migration. So I want you to pay attention to each of these words. Really important to understand that the movement of the immune cells is constant. It doesn't stop in between. It's just an ongoing process all the time. It's also highly regulated. Where the immune cells go from one place to another is determined by several factors that we'll learn today. And each of these factors determine where the cell is going to go and where it's going to end up. And there is a purpose behind where these immune cells end up or they move from one place to another. So few definitions before we keep going. We'll be mentioning these terms here and there, so it's important to know what I'm talking about. So migration and recruitment are used interchangeably. It's the general process of leukocyte movement from blood into the tissues. So these can, be used interchangeably. Another definition to know is homing. That's the migration of leukocytes out of blood circulation into a particular tissue or site of infection. And then recirculation is the ability of the lymphocytes to repeatedly home to secondary lymphoid organs, reside there transiently, and then return to circulation. So for those of you who just joined, we talked about how the immune cells move constantly, and there is a purpose behind their movement, as well as that movement is pretty highly regulated. And some of the definitions are about migration, homing, and recirculation. So when we look at where these cells move from one place to another, it's important to know that the type of cell and the stage of the cell or the phase of the cell determines where they move. For example, the myeloid cells, so neutrophils, monocytes, other myeloid cells, they can migrate from the circulation to the site of infection and tissue injury. That's where they end up and cause the inflammation. Versus naive lymphocytes, T or B lymphocytes, these migrate from their location of where they are produced to secondary lymphoid organs, and they continue to migrate through those secondary lymphoid organs. And then once they become effector or memory lymphocytes, especially the T cells, they migrate into the sites of infection and tissue injury to provide the cell-mediated immunity. Right, so few principles of migration. So for the cells to migrate, there have to be certain molecules on their surface. So there is temporary adhesion of some of the molecules on leukocytes with endothelial cells of the blood vessels or the tissue cells. That's how they move from from blood vessels to the tissues or other lymphoid organs. And then the endothelial cells also express adhesion molecules. They also express chemokines. And the expression of the chemokines is increased by certain cytokines that might be released at the site of infection from macrophages or tissue cells. On the other hand, the leukocytes also express adhesion molecules, and they express chemokine receptors. So endothelial cells express chemokines, and the chemokine receptors are on the leukocytes. And then the adhesion molecules are on both, and we'll talk about what types. Start with the discussion of the couple of types of adhesion molecules: selectins and selectin ligands, integrins and integrin ligands. So what are selectins? So selectin's name comes from the word lectin, and lectin is a surface protein that binds carbohydrate. As simple as that. There is the meaning of lectin is that it is a surface protein and it binds a carbohydrate. Selectins are selected lectins, which means that they are carbohydrate binding proteins that are also adhesion molecules. So those are called selectins. Compared to selectin ligands are a different set of molecules that can bind the selectins. As the name says, they are the ligands for those selectins. So what are these? These are sialylated carbohydrate protein groups of Lewis X and Lewis A family. So they have a particular core molecule which is sialylated and fucosylated, and it's present on glycoproteins of various cells. So to remember, I think that's a lot of chemistry, but just kind of making sure that you remember the words sialylated and fucosylated, and those processes of sialation and fucosylation have consequences if those processes are defective. And so the function of selectins and selectin ligands is to provide low affinity adhesion between the cells and the surfaces. So different types of selectins are P-selectin, E-selectin, and L-selectin. Like everything else in immunology, there are a couple of names to the same molecule. So for selectins, it's CD62. And for each type, there is that letter attached to it. So P-selectin is CD62P, CD62E is E-selectin, and CD62L is L-selectin. P-selectin was named that way because it was found in platelets initially, but we know that it's present on a lot of different cells, and it binds to its ligand, Lewis X, on various cells. E-selectin is also present on endothelium and again binds to Lewis X. L-selectin is present on the cells, so neutrophils, monocytes, lymphocytes, and they can also bind to Lewis X that's present on various molecules. Compared to selectins, integrins are a little different. They are dimeric proteins, which means they have two chains. So they are heterodimeric because there are two non-covalently linked polypeptide chains, alpha and beta. And there are 30 different integrins that are made out of 15 alpha types of chains and 7 beta type of chains. So they have an extracellular head that binds to the ligand, and then they have the intracellular domain that interacts with cytoskeletal components like actin and tropomyosin. With those names, you can imagine that helps in movement of the cells, right? So the intracellular domain that binds to the actin tropomyosin will end up in the movement of the cell if the extracellular head is bound to its ligand. All right, so apart from motility, it can also cause change in shape and help in phagocytic responses. So various types of integrins, few important ones mentioned here: LFA1, MAC1, VLA4, and then alpha-4 beta-7. So LFA1, so the second name for LFA1 is CD11A, CD18. MAC1 is CD11B, CD18. There is another one called CD11C, CD18. So all three of them have the beta chain CD18 in them, and then the alpha chain is different for each one. So MAC1 also has another name called complement receptor 3, and then CD11C, CD18 has another name called complement receptor 4. So again, going back to LFA1, LFA1 is on various leukocytes, and they bind to their integrins called ICAM1 or ICAM2. MAC1, which is CD11B, CD18, or CR3, complement receptor 3, present on various leukocytes and attaches, again, binds to its ligand called ICAM1. VLA4 is a integrin that's again present on monocytes and T cells and binds to something called VCAM. So CAM stands for cell adhesion molecule. ICAM means intracellular adhesion molecule versus VCAM is vascular cell adhesion molecule. So these are ligands for, these are various ligands for integrins. Alpha 4 beta 7 is another important one. It binds to MADCAM1, so another cell adhesion molecule, and it's important for gut and gut associated lymphoid tissues. So the cells that express alpha 4 beta 7 can home to the gut. Remember, homing means that the circulation brings them to go into that tissue and reside there. So that's called homing. So ending up in a tissue is homing. And so alpha 4 beta 7 expression on these monocytes or T cells will help them home to gut. All right, moving on to talk about another type of molecule, chemokines and chemokine receptors. So what are chemokines? These are chemotactic cytokines. They are family of structurally homologous cytokines that stimulate the leukocyte movement and regulate the migration of leukocytes from the blood to the tissues. So there are various families of chemokines, and that's dependent on their structure. Their structure includes cysteine residues that are denoted by a letter C. And so various families of chemokines are named after the number of cysteine residues and intervening amino acids. So the CC family, which means that there are two cysteine residues that are adjacent to each other, are beta chemokines, whereas CXC family is alpha chemokines, which means there are two cysteine residues with an intervening amino acid. C family has single cysteine residue, and then CX3C family means that there are two cysteine residues that are separated by 3 amino acids. So the alpha and beta chemokines are the two major categories of chemokines, whereas there are fewer C family and likely only one known CX3C chemokine. So these chemokines, CC and CXC, which means beta and alpha chemokines, they are produced by leukocytes and tissue cells such as epithelial cells, endothelial cells, or fibroblasts. The production of these chemokines is induced by recognition of microbes through various innate receptors and by cytokines, inflammatory cytokines like IL-1 or TNF, IL-17. The CC type of chemokines are also produced by activated T cells, and they are a link between the adaptive immune system and recruitment of inflammatory cytokines. There are further details of some of these chemokines where they are structurally different and how that affects their function, such as ELR motif. So basically a structure right before CC chemokine could mean that they are meant for recruitment of neutrophils. So these chemokines bind to their receptors that are called chemokine receptors. So as you see in the picture, these chemokine receptors are 7 transmembrane GTP binding GPCR superfamily receptors. What does that mean? These are 7 transmembrane, which means they span the cell membrane 7 times, and they have G protein at the end of it that helps in their function. So the GPCR receptors can bind to the chemokines. And when they are bound, they initiate intracellular responses through associated G proteins. What do? They help in, again, cytoskeletal changes, polymerization of actin and myosin filaments, so that they increase the cell motility. They increase integrin affinity for their ligands, which we'll see in a minute here. And once the chemokine receptor, so there is enough chemokine that can downregulate the presence of receptors on the surface. All the chemokine receptors share some amino acid sequence that is required to interact with G proteins. It's called DRYLAIV, so dry leave. So that's particularly important for these receptors to be able to interact with the G protein that is present at the tail end of the receptors. So just like the chemokine receptors also have families similar to the chemokines themselves that bind to those particular receptors. So CCRs, CXCRs, CX3CRs that bind to particular family of chemokines. These are again numbers, CCR1 to 10 for the beta chemokine receptors, and CXCR1 to 6 for the alpha chemokine receptors, and then CX3CR1 is 1 for the CX3CL1 chemokine. The chemokine receptors are present on the leukocytes, and T cells have different types of chemokine receptors on their surface and most abundant receptors on their surface. So few names here. Of course, this is a long list. Believe it or not, ours, when I was a fellow, this list was 3 times longer. I don't know how they came up with, decided that they don't need to have us memorize all of them. So thankfully, this is all you guys get to learn. Unfortunate part is it doesn't make it any easier either. So again, chemokines and their chemokine receptors here, and then of course the chemokines had different names to them that you are expected to know at least some of these. I'll point out some of the important ones, such as CCL2, that's important for monocyte. It does say it helps with mixed leukocyte recruitment. Same for CCL3, CCL5 and 11 and 17. These are important for allergic processes, so allergic cells. Eotexin is CCL11 for eosinophil, basophil, and Th2 recruitment. CCL17 is known as TARC, and CCL5 is known as RANTES. What is RANTES? Bear with me as I name this. It's called regulated upon activation, normal T cell expressed and secreted. And it's a name that you're supposed to know. I don't understand why. And it's pretty interesting to me. It makes me feel that immunologists and scientists are good at finding things, but maybe not so much for naming things. All right, CCL 21, 22, again, CCL21 is important for T cell and dendritic cell migration, CCL22 for NK cell and T cell recruitment, CCL25 and 27, these are important for homing of cells in particular areas. So CCL25 binds to cells that express CCR9 and help in taking them to the intestine. CCL27 is also known as CTAC and is important for homing the cells that express CCR10 to skin. CXCL1 and CXCL8, that's the next table, are important for neutrophil recruitment. So remember, CXCL8 is also known as interleukin-8, and if you hear CXCL8 or IL-8, think of neutrophils. All right. CXCL9 and 10 help with effector T cell recruitment. CXCL13 is another one to know for B cell migration. Same for CXCL12. All right, so those were the chemokines and chemokine receptors. So we want to know, what do these chemokines do? So chemokines help in recruitment of leukocytes. They also help in development of lymphoid organs and regulate the traffic of lymphocytes and other leukocytes through peripheral lymphoid organs. They help in migration of dendritic cells from site of infection to the draining lymph node. So one of the important function of chemokines and chemokine receptors is also something that's called inside-out signaling, which means they help in the integrins from being in low affinity state to going into the high affinity state. So if you look at this figure, here are the leukocytes. They express integrins and they express selectin ligands. They can bind to their ligands or selectins on the surface of the endothelium, and they can also bind to the integrin ligands on the endothelium. When they are in low affinity state, they can bind to them with low affinity, as the name suggests, but when there is presence of chemokines that can help them to have a tighter or high affinity binding to its ligands. So the chemokine, when it's bound, it signals through GTP binding proteins, and there is association of these molecules with the cytoskeletal interacting proteins with the cytoplasmic tails of integrin proteins that cause a conformational change in the extracellular domain of the integrins. So again, to explain the same thing again, when there is presence of integrin, it can bind to its ligand on the endothelial surface with low affinity. But if a chemokine binds to the chemokine receptor on the surface of the cell, that interaction causes a change, conformational change inside the cell, so that there is a change in the cytoplasmic tail of the integrin, which causes the extracellular head of the integrin to bind with higher affinity. So if you look at that broader shape of the integrin, when the chemokine receptor interaction takes place, because of that, if the effect on the cytoplasmic tail of integrin from inside, it will change the outside affinity of that integrin so that there is tighter binding or high affinity binding of the integrin. So that's called inside out signaling. Important concept to remember. And remember, anything in red is for you guys to memorize. Chemokine integrin interaction. Chemokines induce membrane clustering of the integrins. They increase avidity and tighter binding of leukocytes to the endothelium. So just to kind of make it easy, chemokines help in increasing the affinity and avidity of integrin interaction. There are also some atypical chemokine receptors or ACKRs. They do not use the G protein signaling pathway that we just talked about, and they are actually involved in inhibiting or terminating the chemokine responses in the cells. There are 4 known human ACKRs, and they bind most of the inflammatory chemokines with high affinity. Some of them present on the erythrocytes called ACKR1 or DARK. Can be present on the erythrocytes and postcapillary venule endothelium. All right, and then they basically help in degradation of the receptors, internalization and degradation of the receptors, so they can regulate the function of the chemokines and chemokine receptors. All right, I'm going to stop here and ask to see if there are any questions. We went through a lot. We talked about selectins and selectin ligands, integrins, integrin ligands, chemokines, chemokine receptors, and then the inside-out signaling. So any questions? Okay, let's keep going. We'll talk about the leukocyte and endothelial interactions and leukocyte recruitment into the tissues. All right, so we talk about how all these molecules that we just talked about, how do they function? So the first step is, say there is an infection And from that infection site, there are sentinel cells such as macrophages that release chemokines and cytokines. The cytokines go to the blood circulation to tell them that they need help. So the cells that are the first responders now are supposed to move out of the circulation and go to the site of infection where they can help in fighting that infection. So how does that movement occur? So the leukocytes initially have the selectins that can bind to the selectin ligands. So selectins are on the endothelium, and once the cytokines reach the circulation, there is increased expression of selectins on the surface of the endothelium. And then the selectin ligands can now bind to those selectins on the endothelium. This interaction of selectin with selectin ligands is like Velcro. It attaches and then detaches. And so gradually the cell that attaches and detaches looks like it's rolling on the surface of the endothelium. So it's a rolling stop that the selectins are responsible for. And then the integrins can bind to the integrin ligands. So integrin ligands are on the endothelium. So selectins are on the endothelium and the selectin ligands are on the cell. Instead, integrins are on the cell and the integrin ligands are on the endothelium. So those integrin ligands are ICAM, VCAM. So those are the integrin ligands. So integrin ligands can interact with the integrins that are present on the cell. The interaction of integrins with their ligands bring the cell to a complete stop. So instead of selectin binding to its ligand makes a rolling stop, integrins help in cells to come to a complete stop. And that interaction is made even better when the chemokine binds to the chemokine receptor. So it causes the inside-out signaling. So the affinity of the integrin increases. So there is a stable integrin-mediated adhesion of the leukocytes to the endothelium so that it comes to a complete stop. Once it comes to a complete stop, there is transmigration of those leukocytes through the endothelium. It can be between the endothelial cells, which is called paracellular, or it can be through the endothelial cell, which is called transcellular. They use this molecule called CD31 or PCAM, another integrin ligand that can help in that transmigration of leukocytes. Once it comes out of the circulation, it can follow the scent of the chemokines so that cell is going to end up in the site of infection where the chemokines were initially released. So what happens if some of these molecules are not functional? In those cases, if the adhesion molecules are not functional, the cells are trapped in the circulation and cannot move out of the circulation to go to the site of infection. That occurs in what disease?
LAD.
LAD.
Like 1, 2, 3.
All right, so LAD type 1, type 2, type 3. Let's see what happens in LAD type 1. LAD type 1 is caused by a defect in integrin CD18. That's the beta subunit of LFA1, MAC1. MAC1 is also called CR3 or complement receptor 3, and then complement receptor 4. Which is not mentioned here. But the defect itself is in the CD18. So the CD11A, B, or C might still be intact. The defect in LAD1 causes the cells to be trapped in circulation. So the neutrophil count is pretty high because the bone marrow keeps getting the signal so that the neutrophils are needed. So more neutrophils are produced, they are released into the circulation, but they are trapped in circulation and cannot go to the site of infection. So that's the most severe form of leukocyte adhesion defect. LAD type 2 is because of lack of Golgi fucose transporter. So fucosylation is needed to express the carbohydrate ligands sialoase X of E and P selectin on neutrophils. So the selectin ligands are defective in LAD type 2. LAD type 2 is a milder form in terms of immunological features. So LAD1 is the most severe form, whereas LAD2 is more commonly associated with developmental and growth retardation. LAD type 3 is caused by defect in that inside-out signaling that we talked about. So the signaling pathway defect that helps in increasing the integrin affinity is affected because of a defect in a molecule called kindlin 3. All right, moving on, let's talk about migration of neutrophils and monocytes to the site of infection or tissue injury. So now, think about these cells when they are moving from one place to another. The selectins are binding to selectin ligands, integrins are binding to integrin ligands, and then chemokines are binding to their chemokine receptors. And that's what's making these cells move from one place to another. So now we are talking about neutrophils and monocytes and how they move and migrate to the site of infection. So the neutrophils are first responders. They express CXCR1 and CXCR2 on their surface, and they bind to CXCL8 or interleukin 8 that is produced by macrophages at the site of infection. That's where they are attracted. Similarly, monocytes, these are late responders in terms of infection. They express CCR2 and they move towards chemokine CCL2 or MCP1. There are some non-classical monocytes that express CX3CR1 and they bind to the chemokine CX3CL1. Talking about migration and recirculation of T lymphocytes. So that has various features and we'll talk about each one of them. Let's talk about naive T cells. Where are naive T cells? They are produced in the thymus. Once they are produced in the thymus, they are going to enter the circulation. When they enter the circulation, they go to the lymph nodes. And how do they end up in lymph nodes? So the naive T cells that end up in the lymph nodes get into the lymph nodes through the high endothelial venule. When they are in there, they are looking for antigen that is presented to them that they might be specific for. If they do not recognize the antigen, they are going to move from one lymph node, return to the circulation, and then go to another lymph node. And so they can continue that circulation and again, till they come to a lymph node where the antigen presenting cells express antigen that's specific for that naive T cell. Once the naive T cell gets activated by that antigen presenting cell, that activated T cell will be retained in the lymph node for a little longer. That's how they get time to proliferate, to make a clone. There is clonal expansion, and then they differentiate into activated and then effector T cells. Those activated and effector T cells will leave the lymph node through the lymphatics to enter the tissue. So they are then not going to re-enter another lymph node. They are not going to recirculate. Instead, they will go to the site of infection where they can help in fighting that infection. So HEVs, where they enter the lymph nodes, they are present in lymph nodes. The development of the HEVs requires cytokines such as lymphotoxin. All right, so the naive T cells, how do they recirculate, as I mentioned, and let's see what happens in tissue inflammation. There is increase in the blood flow to the lymph nodes in the area of tissue inflammation. That increases the T cells going into the lymph nodes, right? Because the blood flow is more, there are more number of T cells that are going into the lymph nodes. And there is decrease in the egress of T cells into the efferent lymphatics, which means there are more T cells going into the lymph nodes, but less coming out of the lymph nodes. So the T cells are retained in lymph nodes longer when there is inflammation, so that there is higher chance of them meeting with antigen that they are specific for. So the naive T cells, as they enter the lymph node through the HEV, how do they know where they are going? So the L-selectin on the naive T cells help them bind to the ligand on the HEV, which is also known as peripheral node addressin, and there are different peripheral node addressins for HEVs in different tissues. So the HEVs have these particular selectin ligands that are specific for L-selectin. Those selectin ligands on HEVs can differ based on the type of lymphoid tissue, such as in lymph node, that peripheral node addressin can be glycam1 or CD34. In Peyer's patches in the GI tract, that could be medcam1. All right, so that's selectin binding to selectin ligands. The next step is integrins binding to integrin ligands for firm adhesion, and that for T cells is provided by LFA1 integrin. And then chemokines interact with the chemokine receptors. So which ones are important for naive T cells? CCL19 and CCL21. Where are they produced? They are produced in the T cell zones of the lymph node by FRCs or fibroblast reticular cells. So the FRCs in lymph nodes produce CCL19, CCL21. So the naive T cells that express the chemokine receptor CCR7 on their surface get attracted to the T cell zone where the CCL21 is produced, CCL19 and 21 are produced. CCL21 is also produced by HEVs. That's how the T cells enter the lymph node, and then they move towards the T cell zone. So looking at some of the surface molecules on the naive T cells and comparing them with the ones that are present on activated T cells. So if you look at this table here, the L-selectin, CCR7, and LFA1 are present on naive T cells. Compared to activated or effector memory T cells, they have E-selectin and P-selectin ligands on their surface. And they bind to the E and P-selectin present on endothelial cells. The L-selectin on naive T cells compared to that will bind to the selectin ligands on the endothelial cells of HEVs called peripheral node addressin, which is different in different lymphoid tissues. CCR7 binds to CCL19-21, and then LFA1 binds to integrin ligand ICAM1. Compared to that, activated effector memory T cells express CXCR3, and they interact with CXCL10 in the endothelium. Similarly, they can have CCR5 that is attracted towards the chemokine CCL4. And then again, of course, there are integrins on their surface, LFA1, VLF4, that bind to ICAMs or VCAMs. All right, so the figure here is showing the same thing, where the T cells are entering the lymph node, and they are going to proliferate. They are going towards this T cell zone where there is CCL19, CCL21. And once they are there, if they get activated, they are going to go to the peripheral tissue where they're going to have different surface molecules for activated or effector T cells, so that they can interact with another set of adhesion molecules in the peripheral tissue compared to the lymph nodes. How do the T cells get retained in this lymph node for longer time? So there is something called CYP gradient or S1P gradient. So sphingosine 1-phosphate or CYP binds to its receptor called CYPR1. The CYPR1 receptor is on the T cells. So the T cells or any cell that has the CYP receptor will follow the scent of CYP. So the gradient is such that there is higher concentration of CYP in blood and lymph. So when there is excess of CYP, the receptor on the cells gets internalized. Because it's internalized, it continues to flow through the circulation. When there is low concentration, such as in tissues, because there is CYP lyase, an enzyme that takes away CYP gradient. Because of that, there is re-expression of CYPR1 on the naive T cell surface. As it moves through the tissues in that, because there is CYPR1 on that surface, they are gonna, because there is re-expression, it becomes again responsive to the gradient, so it's attracted towards the higher concentration, so that it's going to move through the lymph node or tissues and go towards the blood or lymphatic vessels, so that it continues to move. That's the naive T cells. But what happens when that naive T cell during that course comes across an antigen that it can activate it? If it gets activated, that T cell expresses something called CD69 on its surface that can bind to the CYPR1. Because the CYPR1 is now bound, it is insensitive to the CYP gradient, so that it's retained in the lymph node for longer time. That's where it gets time to undergo clonal expansion and differentiation. Once it differentiates into effector cells, it again loses CD69. Because it loses CD69, it re-expresses CYPR1 on its surface, becomes responsive to the CYP gradient, and it exits the lymph node. So the naive T cells and effector T cells are responsive to the concentration gradient, but the activated T cells, because they express CD69, are insensitive to CYP gradient and are retained in the lymph node for longer time. So this mechanism was used to come up with an immunosuppressant called fingolimod, and the fingolimod is like CD69, where it actually just binds to the CYPR1, so that the T cells are retained in the lymph nodes. They do not go out to cause the inflammation, so they suppress the T cells from being active. So that's one of the ways that immunosuppressant works. All right, talking about recirculation of T cells through other lymphoid organs. So we talked about lymph nodes. Now, for other lymphoid organs, we are just going to talk about the differences. So for example, for Peyer's patches or mesenteric lymph nodes, so the lymphoid tissue in the GI tract, the movement of the cells is pretty similar. So selectins binding to selectin ligands, integrins binding to integrin ligands, chemokines binding to the chemokine receptors. But the difference is that homing to GI tract involves particular adhesion molecules. In case of naive T cells, homing molecule is MADCAM1, that stands for mucosal adhesin cell adhesion molecule. MADCAM1, what does it bind to? It binds to both selectin and integrin. So it binds to L-selectin. So any cell that expresses L-selectin, such as T cells, right, naive T cells, they can go to and home to the gut. Another type of integrin that it binds to is alpha-4 beta-7. That should have been in red, something that you have to memorize. Alpha-4 beta-7 is an integrin that binds to MADCAM1. So that's for the GI tract lymphoid tissue. Another one is spleen. Now we know that spleen does not have any HEVs. There are no selectin or integrin mechanisms to enter the marginal zone and red pulp. So it's a free entry, free for all in spleen. But still, the cells need some help in moving in a regulated fashion so that they know where they are going once they enter the spleen. So the entry is free. But once they enter, the T cells need to go to the pulse or the T cell zone, and the B cells have to go to the B cell follicles. And what helps them is, again, some of the receptors, such as CCR7 binding, directs their entry into the white pulp, and then particularly for the T cells into the T cell zone. Right, now we talked about naive T cells. Now we're going to talk about effector T cells. cells and how they move from one place to another. So remember, effector T cells are produced in lymphoid tissue, secondary lymphoid tissue, where the naive T cells were able to come in contact with cognate antigen and get activated. After activation, undergo clonal expansion and then differentiate into effector T cells. So effector T cells got produced in the secondary lymphoid organs. Once they are produced, what do they express? They have low CCR7 on their surface and low L-selectin. Because of that, they don't move from one lymphoid tissue to another. CCR7 and L-selectin are like basically maps on the GPS that take those cells back to the lymphoid tissue. Because effector T cells are low in CCR7 and L-selectin expression, instead of moving from one lymphoid tissue to another, they will enter the circulation and can go to the site of infection. They can go to the tissues. And the activated endothelium will express particular ligands that will attract these effector T cells. Effector T cells have selectins interacting with their ligands, integrin ligands interacting with integrins, and then some of the particular chemokine receptors that are important for effector T cells to enter the infected tissue. And then, and it's different for certain types of cells. Nothing big there. Tissue-specific homing, we talked about gut homing a little bit, a few more details to that. For chemokines, CCR9 is any cell that expresses CCR9 will be attracted to and bind to CCL25. CCL25 and 28 are present in the gut. So CCR9 binds to CCL25 and CCR10 binds to CCL28. Apart from the gut, GU system as well as lungs also express CCL28. So cells expressing CCR9 and 10 will be attracted to those places. MEDCAM1 binds to alpha-4 beta-7. Can't emphasize more that that's an important factor to memorize. Skin homing. So there are some cells that express CLA or cutaneous lymphocyte antigen. CLA binds to E-selectin. So it's an important factor for the cells to home to skin. So, similarly, there are cells that express CCR4, 8, or 10 that get attracted towards CCL17, CCL1, and CCL27. So look at that. CCR10 binding cells or expressing cells can be attracted towards CCL28, which is in gut, or in skin, which is CCL27. And again, something clinically important is alpha-4 beta-7, and a drug called vedolizumab can be used. We talked about spleen, and we talked about tissue homing, and vedolizumab, that is alpha-4 beta-7 antagonist. And then we talked about effector T cells. Now it's memory T cells. There are two types of memory T cells, central and effector memory T cells. Central memory T cells express CD45RO, CCR7, and L-selectin. So they have CCR7 and L-selectin. Guess where they're going to end up? They're going to end up in the secondary lymphoid tissues where they're going to reside. Effector memory T cells, on the other hand, have low levels of CCR7 and L-selectin. So instead of going to the secondary lymphoid tissues, they're going to end up in the peripheral tissues where they're going to reside. They also express CD45RO on their surface. And then the last topic is migration of B lymphocytes. They are produced in bone marrow, and immature B cells move into the circulation and end up in spleen, where they mature into naive follicular B cells. Spleen has CXCR5. Spleen has CXCL13 in the white pulp in the B cell area, where cells expressing CXCR5 can be attracted to. So CXCR5 expressing naive B cells or immature B cells will end up in the white pulp and bind to the CXCL13. They mature into naive follicular B cells, and they can again recirculate to the lymph nodes. They enter in the lymph nodes. They enter the lymph nodes using CCR7 on their surface. Because of that, they bind to CCL19 or 21 to enter the lymph nodes through the HEV. But then they also express CXCR4 on their surface that helps them to bind, go into the lymph nodes. They also express CXCR5, which is attracted towards CXCL13 that is present in the follicles, and the CXCL13 in the lymph nodes is produced by follicular dendritic cells. In the Peyer's patches, again, the CXCR5 helps them bind to CXCL13, and they also express alpha 4 beta 7 that helps them to bind to MADCAM1 and home to the gut. Once they are activated and differentiated into plasma cells, these plasma cells can home back to the bone marrow and tissue sites where they reside and produce antibodies. Follicular B cells in the spleen migrate to the marginal zone and then carried through the red pulp and into the circulation. The CYP1 deficient B cells have diminished ability to leave the spleen. So those are the some of the B cells that can reside in the spleen. Various type of plasma cells, such as IgG secreting plasma cells, will home to the bone marrow, and the VLA4 and CXCR4 bind to their ligands, so VCAM1 VLA4 and CXCL12 CXCR4 interaction will help them to stay in the bone marrow. Whereas IgA secreting plasma cells can be present in the mucosa. So these plasma cells will express alpha 4 beta 7 that helps them bind to MADCAM1 and express CCR9 and 10 that helps them bind to CCL25 and 28 on mucosal endothelial cells. There are some IgG secreting B cells that are present in chronic inflammatory sites. They express CXCR3 and VLA4 that bind to VCAM1 and CXCL9 and 10 found on the endothelial cells of chronic inflammatory sites. All right, so that's the last slide here, just summarizing what we talked about. B cells moving from lymph nodes to HEVs and then into the B cell follicles, and then the plasma cells going into the bone marrow for IgG secreting plasma cells and mucosa for IgA secreting plasma cells. Couple of questions here. Migration of leukocytes out of the blood vessel into the tissues mainly occurs in which type of vessel?
Should be venules.
That's correct, venules. And then which of the following is correct for naive T cell migration? Did you guys read that?
I think this one is A. They go from the blood to the lymphatics through the high endothelial venules and then back.
That is correct. All right, those were the two questions. And then the next lecture is Jeopardy for our first 3 chapters.